Good morning, everybody, and you're very welcome to our Signpost Series webinar this morning. My name is Andy Boland, and I'm going to be your host for the next hour or so as we talk through and discuss another issue of farming and the environment. I'm delighted to be joined this morning by three colleagues, two from the Agricultural Catchments Programme, Dr. Bridget Lynch and Dr. Ognjen Zurovec, and both of them are going to give us an update on the Catchments Programme. But before that, it's been a very busy week. Both of you are joining us from Clonakilty this morning because it's the end of your Agricultural Catchments Open Week. And before we begin the presentation, maybe you might like to give us a brief rundown on, well, what you're doing yourselves within the programme, but also the week and how it went for you.
So I'm a research officer. I'm a research agronomist in the Agricultural Catchments Programme and in the wider Environment, Soils and Land Use Department. I'm based in Johnstown Castle. This is the last day of our Agricultural Catchments Week. So it started last week with a webinar by my colleagues Greg Melander and Jason Galloway. And during the week, we've had a number of podcasts, online articles have been published on Teagasc Daily. And we have our webinar now again here, and then we will finish up with an open day, a farm walk at our Timoley catchment at the outlet point. So that starts at 11. So myself and Ognjen are here in Clonakilty Agricultural College. They're facilitating us here to do the webinar, and it's a lovely sunny day in West Cork. So we couldn't ask for better, I guess, for our open day to finish up the week.
Okay. We're also joined by Tom O'Dwyer. Tom is the program manager for the Signpost Programme, and Tom is going to keep an eye on the Q&A and help facilitate the questions as we after Ognjen and Bridget's presentation. Morning, Tom. Thanks for giving us a dig out.
That's okay, Andy. You're welcome. I'm interested to hear the presentations from our two colleagues, Bridget and Ognjen.
Ognjen, you might tell us a little bit about yourself and what you do, what work you do within the ACP.
Yeah, good morning, everybody. Thanks, Andy. Yeah, my name is Ognjen Zurovec, and I'm a soil scientist in the Catchments Programme. So, well, it was a busy week. Yeah, I've been in Clonakilty here since Wednesday. I have to do some of my regular soil and water sampling, as you will see in my presentation. And yeah, so we're here for the webinar now, and we'll be heading off to our open day later, as soon as we're finished here. And yeah, we're very lucky with the weather, and yeah, looking forward to it.
Okay, well, thank you both very much for taking the time out of a busy week to talk to us. I'd just like to remind people that our series is brought in conjunction with Dairy Sustainability Ireland, Food Drink Ireland, SkillNet, and the National Rural Network. And a recording of today's presentation will be made available on our website, and we also do a podcast version wherever you wish to download your podcast. So, Bridget, you might like to begin and get the ball rolling and begin your presentation.
Okay, so I assume you can see that okay. So I'm going to give an update on the gaseous emissions part of the programme on behalf of myself and MacDara O'Neill, who was the gaseous emissions research officer who worked on the programme until recently. So just in case any of the viewers this morning weren't tuned in last week, just a quick rundown on the Agricultural Catchments Programme. It was established in 2008 and has been fully funded by the Department of Agriculture since then through 4-year funding cycles, and we're currently in the 4th phase of the programme. So it's a unique programme in that it combines both research and knowledge transfer, and our staff are made up of technicians, technologists, research officers, in addition to advisors and a knowledge transfer specialist. So we have 23 staff across 6 agricultural catchments, and we engage and have 300 farmers that we deal with in those catchments. It collects, both biophysical, so that's the soil science and water quality and gaseous emissions data, as well as having a socioeconomic appraisal of the programme and the policy that we review. It's a focus point for catchment science knowledge transfer and for water quality, and also gaseous emissions knowledge transfer, and that's evident, I guess, in the drive that we have this week with our Agricultural Catchments Week. So the programme was set up to evaluate the nitrates action programme in addition to the nitrates derogation, and I guess it has evolved to be a wider policy evaluation programme. The programme, I guess, has evolved over the phases, and I guess in the current phase, we've probably seen the largest kind of expansion of the baseline biophysical data collection, in that it expanded the remit of the programme to include gaseous emissions, so greenhouse gases, ammonia and carbon sequestration, in addition to adding to the soil science remit of the programme by looking at soil solution monitoring for nitration phosphorus. And Oggie's going to collect that, or going to focus on that in his presentation. So I guess the reason for the expansion and the inclusion of the gaseous emissions in what is a water quality focus programme in the first 3 phases is to see, I guess, what regulations may be brought in to improve water quality or gaseous emissions, if they work in tandem with each other, or if they're perhaps antagonistic to each other. So this is, I suppose, the core baseline data collection that we do in the programme. So it works on the basis of tracking the nutrient transfer continuum. So the source of nutrients, so nitrogen and phosphorus, the mobilisation of those nutrients, the transfer of them through the landscape, and the delivery of those in the river, and the impact of that delivery and those nutrients on the freshwater ecology. So where does, I guess, the gaseous emissions side come into, or how can it complement the current measurements that we do? So the management data, or the information that we know with regards to the imports of nutrients and also carbon as well, is ongoing through LIPIS data from the Department of Agriculture, but also through our data recorders and our farm advisors. And that's important, I guess, when we're looking to try and understand imports and exports of carbon, but also ammonia as well. We do a soil sampling campaign, and Oggie will expand on this later, every 4 years. They're agronomic samples, but we've been able to go back and analyse them for carbon, nitrogen, total carbon, nitrogen, and also organic carbon. We have a MET station in all of the catchments. Transfer pathways have been identified throughout the programme. And then with regards to our monthly snapshot samples and the high resolution monitoring at the outlet, we have been measuring TOC at the outlet for the last 4 years. So we can potentially, I suppose, in addition to nitrogen and phosphorus and looking at its transfer continuum, we can also look at the carbon transfer continuum and look at carbon losses, or hydrological carbon losses. So this slide summarises the ongoing work on the gaseous emissions side. So there's 5 edicovariance towers that have been deployed across a number of the catchments, and that started in late 2020. We have analysed the first soil sampling campaign, agronomic soil samples for total carbon, nitrogen and organic carbon. And we have also analysed the most recent campaign that was done in phase 4, and we're able to look at the change in soil organic carbon in the first 10 centimetres as a screening tool. We have begun the soil profile pit digs to find out what the bulk density and percent organic carbon is, where the edicovariance towers are situated. And we do that in tandem with other similar programs like the Signpost, to make sure that, I suppose, all programs are working to the same methodology. We're also measuring active ammonia in the atmosphere in Castledockrell, which is hypothesized to be our low ammonia site, the Castledockrell crop site, and then also Ballycanoe, which is a dairy grazing site, which is hypothesized to be a high ammonia site. And then there's other co-located equipment. So the ISMON program, which has deployment of the Cosmos in situ soil moisture array at various locations across the country, but there's one of those co-located with the tower in Ballycanoe and Craigduff. Through the Terrain AI program, there's been extensive drone surveying done at the Castledockrell grass and crop sites. And in addition, there's soil auto chambers installed at the Castledockrell sites that can measure carbon dioxide as well, and to compare that to the flux data that we get. So I suppose in the update today, I'm just going to concentrate on the ecovariance tower work and look at some of the outputs from that, some of our initial observations. So the towers were placed in 5 different locations. So Craigduff is our extensive grazing site. So this is where we've grassland that's extensively grazed by sheep and beef animals that are co-grazing. Timoleague is our intensive dairy grazing / silage site, and that's a freely draining soil type. And the tower is located on the milking platform on that farm. Castledockrell, we have 2 towers. So we've one in a crop rotation site and one in a zero grazing / silage cutting site, grassland site, and that's freely draining. And then in Ballycanoe, we've an intensive dairy grazing. It's situated on a paddock in the milking platform, and that's a poorly draining catchment or soil type there. So I'm just going to concentrate on, I suppose, the first 4 there: Craigduff, Timoleague, Castledockrell, crop and zero grazing in this presentation. And just to say, I suppose the site selection for these towers, there's quite a lot of work goes into site selection as to where they're going to be situated. Not all sites, fields or farms are suitable. You need a flat terrain. You need to have a southwesterly facing fetch. We need management information to ultimately be able to do a carbon balance. And then we also need, I suppose, most importantly, it should be top of the list, farmer engagement to allow us to deploy these pieces of equipment onto privately owned farms. So these 5 towers, and people may be familiar with the National Agricultural Soil Carbon Observatory, or NASCO. So these 5 towers are part of that. So there's 30 flux towers being deployed as part of NASCO, and our 5 towers will feed into that network and into that data as well. So similar objectives, I guess, across both programs, to investigate agricultural land use and the management inputs, and I suppose what impact that has on annual estimates, and what are the drivers of carbon gain and loss. So there are, I think, 5 more of the 30 in total to be deployed, and the target is to have that done by the end of this month. So this is the equipment that you would see or maybe have seen maybe on some of the research farms or out on some of the commercial farms. So the ecovariance tower method of measuring carbon dioxide is what we call a micrometeorological method. So in addition to having a carbon and a water vapor analyzer, which is the case in the towers that we have, you also have a number of, I suppose, equipment that measures the immediate climatic conditions, because they're very important, I guess, explainers of the carbon dioxide fluxes that we observed. So there are a number of other analyzers that you can get and attach to the towers as well. So you can get a methane analyzer and a nitrous oxide analyzer. We just have the carbon dioxide analyzer on our towers. So they operate by measuring the fluxes of carbon dioxide, so what is photosynthesized from the atmosphere by the plants, in this case it's grassland or crop production, and then what is respired by that agricultural ecosystem. And the difference between what is photosynthesized and what is respired is what we call net ecosystem carbon dioxide exchange, or NEE is what I'll refer to it throughout. So the towers or the analyzers, they take 10 measurements per second, 365 days a year. So highly, highly automated. And as I said already, the biomet sensors, they, I suppose, help us identify the drivers of net ecosystem exchange. So I'm just going to explain maybe some of the terminology that you're going to see in the graphs in the next couple of slides. So the unit of measure in the y-axis is fluxes of carbon in micromoles of carbon dioxide per meter squared per second. And we have days of the year on the y-axis. This black line, horizontal line here, just indicates where 0 is sitting. And the blue line that you'll see in all the graphs is RE, or it's ecosystem respiration or respiration. The red line is what's called gross primary productivity or photosynthesis, and that's the red line. And then the difference between these two is our net ecosystem exchange, and that'll be represented by a green line in all the graphs. So if the green line or net ecosystem exchange is below 0 and it's a negative value, that means it's a sink of carbon dioxide. If it's above the line, that means it's a source of carbon dioxide. So first I'm going to show you our arable site. So this data is from the 20th of January through to the 1st of November 2022. And it's a winter wheat crop that was grown that year, or last year. And we have N1 represents the first application of nitrogen, N2 is the second application of nitrogen, and then we have the harvest as well that occurred later in the year. Underneath then we have biomass, and we have the flower is represented by the red line. The green serrated line is the leaf, the root is the blue line, and then the purple is the stem of the crop. So it's important that we take biomass samples throughout the year to understand what the carbon uptake is in the biomass samples. So what do we observe? So we observe greater net ecosystem exchange corresponding with increases in leaf area index. So we saw uptake throughout the year, which peaked corresponding with the peak in leaf area index, which corresponds to about day 140 down here. We had sustained carbon uptake during grain filling. However, ecosystem respiration exceeded net photosynthesis about 3 weeks out from harvest. And obviously, as the crop starts to mature and become close to harvest, you've less green leaf material or green material, and there's less photosynthesis. And then after harvest, we can see that there was a small carbon uptake recorded with the volunteer growth that came there afterwards. This is our our dairy grazing site. So what we've got up here is the flux footprint, or the contour map showing the flux footprint. So the tower is situated here. You can see the contour lines have numbers on them. So you can see this one says 80%. That means that 80% of the flux data that we're recording is explained by the activity of that area. And we also have the windrows map here as well, just for your information. So in this map, we've got a number of harvests, obviously, because it's a grazing and a silage cutting site. So we have the harvest throughout here. You can see that well, first of all, the fluxes that we observed, the variation and the magnitude are what we would expect in a grazing situation, rotational grazing situation. So carbon uptake progressively increased to a maximum series of cuts. So you can see that we'd uptake, I guess, right throughout the grazing season. And then that started to decrease and become a net source as we moved towards the latter end of the grazing season. Here, this where we can see the net ecosystem exchange above zero. So this also coincides about July-August period. And we might remember last year we had a drought period around then that stretched into September, also having an impact. This is our zero grazing site, zero grazing or silage cutting site. So again, the contour map showing the flux footprint and our windroses. And here again, we have the various harvests or defoliations or grazings or silages represented by the black lines indicated there. So the flux magnitude coincides with the cutting period, and the rate of carbon uptake was lower after defoliation. So I guess after an offtake, and typically I suppose farmers zero graze at higher covers than they would graze at. And obviously after silage cuts, there's not a lot of green leaf material there to photosynthesize, and the rate of carbon uptake is lower after the defoliation. We also saw an impact, I suppose, of time of year and also probably the drought as well between harvest 4 and 5 here, where you can see that the net ecosystem exchange green line is above zero and becoming a source there. This is our extensive grazing site. So again, black line representing zero. This site is continuously grazed by cattle and sheep co-grazing together. So we don't necessarily have harvest lines in this, as it's been continuous. It's in a continuous stocking system. The max daily photosynthesis is generally higher compared to the arable or the dairy grazing sites. We see that the trends of net ecosystem exchange, you can see that it's a much more jagged line, if you like. So it fluctuates widely. And I suppose the drivers of that could be weather or management. And similar, I suppose, to the arable and the zero grazing sites, the net carbon uptake slows down at about a day 300. So you can see that net ecosystems exchange is running along there underneath the line. And then as we reach towards the end of the year, it starts to become a source, or it starts to slow down. The respiration rate was higher compared to the other sites. So there is perhaps a carbon dioxide flux source below ground. So it could be, you know, this is a permanent pasture. It's a lot more diverse compared to, I suppose, the newer pastures that you would typically have on a dairy grazing or around the zero grazing site. So perhaps there is more respiration coming from the root network or the microbial population in that site. So it's quite interesting when you compare it to the other intense grazing grassland sites. So to summarize, net ecosystem exchange at the arable site is regulated by crop development, and that would tie in with other observations from work to date. Cutting and grazing frequency observed to influence net ecosystem exchange at the grassland sites, and the rate of carbon uptake was lower after defoliation. The recovery period seems to take longer as the season progresses, and can be limited by the weather conditions or the drought conditions that we saw last year. The extensive grazing site, the trends of net ecosystem exchange, they fluctuate widely, and the drivers of that, I suppose, need to be examined. So whether it's weather management, the underlying soil processes and respiration, I suppose that work continues. And look, these are initial observations. We need more years of data. These towers are being deployed for the long term. So we need longer data sets and greater continuity of the data before we can definitively, identify management and weather effects or drivers of what we're seeing. And look, the work continues. So what I showed today is output from just the flux towers. We need to couple that with management data in order to do a full import and export of carbon to the system and to come up with a carbon balance of tons of carbon per hectare per year. The baseline soil organic carbon profile pits are continuing this year. And I suppose this ties into the new Teagasc Climate Centre roadmap, where by Q1 of 2027, the target is to have Irish-specific soil carbon sequestration emission factors determined by then. So that's it from me. And I can stop sharing and hand over to Oggy.
Thanks very much, Bridget. And just before Oggy starts or gets going, could I remind people again to send in the questions? There's loads of questions coming in. So Oggy, are you ready to share?
I am. Just
Yeah, good.
Okay, can you see my presentation?
Yeah, very good.
Okay. Okay, so yeah, once again, hello everybody, and I'm going to give you an update on the latest research, soil research in the Agricultural Catchments Programme. So Bridget gave a good overview of all our activities, like from all sides of the Catchments Programme, and she also mentioned that we expanded our activities to first the greenhouse gas emission side, which she explained in detail. And in terms of soils and soil research, the significant expansion came in the context of so-called soil solution monitoring. So just before I move on to the main activities in relation to our soil solution monitoring, just to say a few brief words about, I'm sure most of you are aware about the nitrogen use efficiency on the farms, and that NUE or nitrogen use efficiency sits around 25%, like on a typical grassland dairy farm in Ireland, which means that up to 3/3 or 75% of nitrogen is either fixed in soil or lost somewhere in the environment, usually as gaseous emissions like ammonia and nitrous oxide. And what we're going to focus on, and what will be kind of the main point of this presentation, will be focused on nitrate leaching. So all the nitrogen which leaves the root zone is, since it will be out of the reach of the root zone and will never be taken up by plants again. So that nitrate is very mobile and will move with the groundwater, with the soil water eventually to reach the groundwater, and from there it could have an impact on our water bodies. So we already have, in most of the catchments, we have a groundwater monitoring network. And with the soil solution monitoring, we are kind of adding one piece to the puzzle there to estimate or to measure how much nitrate will leave the root zone or is leaving the root zone, and then combine it with our groundwater and high resolution water monitoring to kind of get the better picture and knowledge of the nutrient cycling and the processes happening since after the nitrate leaves the root zone on its way to groundwater and our surface water. For the soil solution monitoring, we picked the Timolee catchments as a case study, and the reason why Timolee catchment is located in West Cork near Clonakilty, and the reason why we picked Timolee catchment is that it represents one of the most intensively farmed regions in Ireland. And another thing is because, as you can see on the soil map here, see that the catchment is dominated by so-called canvas soils. You'd know them better as brown earths, which are well drained. So there is a risk there because because the soils are freely drained. There is a risk of nitrate leaching on these soils and in this area, which could have an impact on water quality in the region. Another fact about Timoleague is that I already mentioned it's one of the most intensively areas in Ireland. As you can see here, the dominating land use in the catchments is it's mostly or almost completely under grasslands. And as you can see on the right figure here, that it's also highly stocked, and most of the farms or the areas here are under derogation. So what was the selection process for the sites we've chosen to install our equipment and measure soil drainage? One of the main points we want to see is the difference between potential for nitrate leaching between a derogation and non-derogation farm. And we started, as you will see later in the presentation, we started with 2 sites, one in derogation and one in non-derogation, but then we decided to expand further. We wanted all the sites to be on the same soil type so they can be comparable. So we picked the soil type you've seen in the previous slide, which was the brown earths, which are dominant in the catchment. And yeah, we also, in order for the sites to be comparable and be in the similar conditions, so we picked the sites which were similar in kind of physical setting, which be topography, altitude, and slope. So you see the selection process there, and then at the end, you see the area which we had at our disposal, which met all those 3 requirements. And then within this area, we had to choose the suitable sites for what we wanted to accomplish. So that brings the other 2 points for the selection process. We wanted variability in farms in terms of the organic and loading. So we'll have different farms, as you will see in the next slide. And we also wanted them to differ in grassland management. So some of the sites would be like in part of the milking platform. Some of them would be combined kind of silage and grazing. And we also have one site which is zero grazing. So here you can see the overview of the sites, and you see that we have 4 sites in total, and that they differ in terms of their management. So for the non-derogation site, we picked the low input pasture, which is on the mix farm. And then the other 3 sites are in derogation, but they differ in their management. So one of them is intensively managed grazing paddock, which is a part of the milking platform. And the other site is used for silage, first cut silage in the spring, and then it's in the grazing rotation for the rest of the year. And then, as I mentioned earlier, we have a site which is used in zero grazing system and for silage. So, and you can see in the table below there, organic and inorganic and inputs. This is typical throughout the year. We record all these information, that we keep the record of everything which is applied, all the organic and inorganic fertilizers applied in the field. And we also estimate the organic and input based on the herd size and the amount of time the animals spend grazing in the field, like per grazing period, and then total throughout the year. So what you see here, the total and inputs, they're not, you'd usually see them represented per farm. These are the estimations only for that specific field. We're talking about where the lysimeter site, where the soil solution monitoring site is. With the exception of number 4 here, you see it marked. We still have incomplete data for that site. I think this is up to August or something like that in last year. So I mentioned already, we started with initially with 2 sites, and they were established in autumn 2021. And we've been trialing 2 different sampling methods, which you will see on the next slide. And once we accomplished our main objective, which was to monitor soil solution on derogation and non-derogation farms, which was also the requirement of and something which the European Commission asked and required in terms of nitrate derogation. So once we fulfilled that objective, we decided that, yeah, we want more, and we want to move one step further and add more sites which differ in management. So we added 2 new sites in September last year. And as you can see on the pictures here, like it's a very like the lysimeters and we use, and they're put underground, and it's a fairly aggressive and destructive process, and therefore like it takes time for the soil structure and the root zone above the lysimeter to kind of recover and is back into the natural conditions. So that's something you cannot rush, and that's why we expect, although we started 2 years ago, we expect our first kind of full scientifically sound and then comprehensive results, at least from the two sites in which were established in 2021. We'll have the full comprehensive results in summer 2023. The drainage season, it has been a fairly wet year, and the actually drainage season is still ongoing. I was on the sites last 2 days and I was still taking samples, and there's still a drainage collected in the lysimeters. But yeah, it's expected that the drainage season will be over probably in the next week or two. So, and that will give us the time to sit down and analyze the results and have something to report later in this year. So how do I keep talking about the sites and the lysimeters? So how does what you see on the right is the kind of typical layout of one of our soil solution monitoring sites. So you see that each site consists of 6 so-called wick lysimeters. These are those big steel rings you've seen on the picture on the slide before and on the picture on the left. And then we also have 3 clusters of 4 ceramic cup samplers per site, which brings up to 12 samplers in total. So we have 18 samples coming from each field every time when we take the sample. And we've chosen 2 different ways to take samples and to monitor soil solution. And we're still in the process of figuring out which method works best in the conditions we have. And it's also important to mention, and which is unique for this type of research, is that all sites are actually located on the actual farms, and they are under realistic farming conditions. And it was a big challenge to actually have the whole setup underground, so it doesn't interfere with the farming operations, like the passing of the tractors, like cutting the grass and grazing and so on. So you will see that we developed our approach was to keep everything underground, and you see that the only thing kind of sticking out from the surface are those access caps in the top right corner. You can see them. And so, yeah, the whole setup is underground. And we are also, very lucky that we have the support of the farmers in the region, and that they allow us access to their land, and they tolerate our presence every week or fortnight during winter when we come to sample water from the lysimeter. So, yeah, we are very grateful for that. So what is done with the samples, and how often do we sample? So usually, it's the drainage season in Ireland usually starts in early October and lasts until May. So that's when the rainfall is higher than the evapotranspiration. So the excess water is being drained below the root zone, and our lysimeters are located just right below the root zone, and 30-40 centimeters below the root zone, and they go up to 1 meter. So we collect all the drainage up to 1 meter depth. And so when we are on site every week or fortnight, we take the water sample, we pump out the sample with the vacuum pump, we measure the volumes, and using those volumes we can record the amount of drainage, and then we take subsample from that sample and bring it to our water lab in Johnstown Castle to be analyze for nitrogen and phosphorus. And what is also important in this monitoring is to have the constant data recording and to be in constant contact with the farmers and make sure that we record all the major farming or events, like mainly like fertilization, either organic or mineral fertilizer, and to record all the grazing events on the site with sites which are grazed. So yeah, that would be all in terms of lysimeter or soil solution monitoring. And then the second thing I would like to talk about as a part of this presentation, which I hope many of you will find interesting, is the soil fertility trends in some of our catchments. So in this case, it's before catchments where we have the data for 4 years and where everything is analyzed and prepared. As you heard from Bridget, ACP has been running since 2008, and I think the first soil sampling started already in 2009. So we have roughly 12 to 13, 14 years of sampling by now. And although it's a relatively short period, a lot has changed in the Irish farming sector since the ACP has been established. The most important thing, of course, would be the abolition of milking quotas, and also many new policies and regulations aimed at promoting sustainable practices have been introduced since. So since we have a large data set of soil samples, and we were interested to see what was the change in soil fertility during this period. And we selected these 4 catchments. So 2 of them are in Wexford. Our Dunleer catchment is in County Louth, and then, yeah, already mentioned Timolee catchment in Cork. How does our soil sampling soil sampling survey or audit looks like? So the agricultural area of each catchment is divided into defined sampling units, which are roughly around 2 hectares. So if there is a field bigger than 2 hectares, then we separate or split it into the subunits and subfields, and then you see how the sampling units look like on the figure on the right hand side, and then that brings up to 5 or about 600 samples on average per catchment, repeatedly taken from each sampling unit every 4 years. And that makes it unique in a sense that, yeah, we're able to track the changes in soil fertility from almost every field in the catchments. And that's unique, and that's why this data set is very valuable, and it's unique not just for the catchments program, but like nationwide. I don't think they're although the areas that covered by catchments are about 10 square kilometers, like they're relatively small in size, but the detail we have is very good and very interesting. So, and all these samples are, so what we're analyzing the samples for are our main soil fertility indicators, which would be pH, pK, and lime requirement. We do sample. Sometimes we sample them for micronutrients, or as Bridget mentioned, all the samples, because we archive them, so all of them have been reanalyzed for carbon content as well. Now, I could overwhelm you with lots of figures and graphs showing the changes in soil fertility in each catchment and for each nutrient, but yeah, instead I prepared this simple figure, and I will just point out the most important points and the most important things when it comes to changes in soil fertility trends over the last 12 years or so in the catchments. So in general, we see a small increase in soils with good overall fertility, which is now, of course, a positive thing, and it shows that we are slowly but steady going into the right direction. And probably one of the most positive things is a very positive trend in terms of soil pH. So there has been a 20% increase in soils above pH of 6.2 over since the first time the soil samples were taken from the catchments. In terms of K, usually, 2/3 of the soils are well supplied with potassium, which is also a positive thing. And then when we're talking about phosphorus, we have there are there are some positives and some not such good so good things like in terms of phosphorus. So what we can point out here are kind of two main concerns in relation to soil test P content, and the first one would be environmental, where nearly 20% of our soils are P index 4, which means the excessive soil P content, and that presents that's not really positive in terms of when we're talking about the environment. But we also have an opposite trend, that there has been an increasing there are increasing areas, and there's like the P contents actually decreasing in some of the catchments, and more than 60% soils are also but they are below the agronomic optimum. So we have two different sides of the story here. We have the fields which are with excessive P content, and then we have substantial areas which are deficient in phosphorus. And then, I will just conclude with saying that, in terms of phosphorus, there's significant scope to correct the P balances on farms with the better nutrient management. You see the typical dairy farm on this figure, and you see that most of the phosphorus is located within the milking platform. And so there is scope of improvement to, especially when we're talking about organic manures, to kind of bring the balance to the areas away from the milking platform, and in terms of productivity and in terms of environment. So I will stop here, and thank you very much for tuning in.
Thanks very much, Augie. So Bridget, thank you. If you might like to bring back on your camera, and Augie, thanks for stopping to share. Thank you both very much. Look, we've a good few questions coming in, so we haven't got a whole lot of time left, so we'll try and get through as many of them as quickly as we can. Just the first thing that struck me really, Bridget, when you look at both your presentations, there's a lot of new kit, extra kit, invasive kit gone onto farmers' land. I mean, have you had? I mean, has there been issues with that, or has everybody welcomed you with open arms, or what's been the reaction when you turn up with all this stuff to land on a farm?
Yeah, well, I suppose there's a good bit of work goes in before we land down with all the gear, Andy. So when Makdara and Zaid were the research officer and technologists that started the deployment of the towers and the catchments, they would have looked at sites that were suitable. So not everywhere is suitable. So they did a good bit of sort of decision support tree, if you like, or analysis on site selection. But then they would have sent out an email via the catchment advisor, to see who was interested, firstly, before knocking on doors. So the farmers would have been asked who would have been interested to speak to the researchers, and see who might be interested in hosting. And so it was voluntary in a way, from, I suppose, those sites that were suitable.
Yeah, but it's still it's a lot of stuff on your farm, And
it's a big ask, to be fair, and they need a power source, so you need a connection to electricity, and you need to fence off a bit of your milking platform, and that's precious. So we're very appreciative.
Always grateful to people who who do this for us, really. Just to pull a couple of questions together, Bridget, mainly for yourself. Now, I say, what can farmers do to improve carbon sequestration, and those figures that you showed us? Is there any rate per acre that you could give a guy or per hectare as a guide, or what could farmers do to improve rates of carbon sequestration?
Yeah, so I think what we see both across the grassland and the crop sites from those observations is that leaf area and leaf area index is going to promote photosynthesis and carbon dioxide capture. So if you think about, I suppose, avoiding ploughing if you can, both by machinery and by animals. So try and avoid damage to the soil surface, during difficult grazing times of the year. Look at minimal cultivation techniques for establishing grassland and for cropland, avoiding fallow periods, so trying to enable the establishment of volunteers after harvest or for cover crops, look at cover crops. And then, look at mixed species. Beyond then the fields themselves then is to enhance hedgerows, quality, quantity, and so on. So look, the mineral soils, I suppose they have the potential to sequester 1.5 to 4 tonnes of carbon dioxide per hectare per year. So there is the potential there.
Yeah. And just finally, before I go to Tom, most of your flux towers are obviously measuring above the ground. Is there any relationship between carbon in the soil, or did you look at carbon in the soil below the ground, we say from a tillage to a grassland, intensive grassland to an extensive grassland? Is there soil organic carbon, or is there any?
Yeah, so
that came in as well.
Yeah, so one of the initial things that the team did was to look at the soil organic carbon in the archive sample. So it's just in the first 10 centimetres, but they were able to look at grassland, permanent grassland that was converted to tillage, and then tillage converted to grassland, or to look at the land use of that going from phase 1 to phase 4. And they definitely would have seen that there is a positive change in soil organic carbon. It's only the first 10 centimetres when it stayed in grassland, and where we saw more organic manures being applied in the catchments as well. And they would have found that if there was grassland lay as part of tillage rotation, that would have decreased decline in soil organic carbon in the arable sites.
Okay, Tom, over to you.
Thanks, Andy. A couple of questions in for yourself, Bridget, and then Oggy. So maybe I'll stay with you, Bridget. So the first one is whether your results provide any indication as to whether intensive or extensive grazing systems sequester more carbon.
Yeah, so I suppose intensive grazing systems, you have more return of an even return of animal excreta to the soils. So there are some positives around that. Extensive grazing, obviously you've less chance of poaching taking place, or there's less reseeding as well. Probably that's going to allow ploughing to take place. So look, I think there's pros and cons of both. We have the intensive grazing sites, and we have the extensive grazing site in Craigduff. So when we pull together the imports and the exports of carbon overall and do the carbon balance, we'll be able to answer that better.
Okay, all right. And a second question then from Orly. How can we compare two systems with or without grazing animals around the towers? And I think you have that. I think you had a silage zero grazing system, and you had the tower with intensive grazing and extensive grazing. So can you make that comparison, Bridget? Is that fair?
Yeah, I suppose they're not completely directly located at the very same site, but yeah, look, that's been captured across the 30 network. Like some will be grazing, some will be cutting sites, and the towers can pick up on the animals when they're there around the sites as well. So it'll pick up them and also the machinery as well when they're harvested. So we've the ability to do that from what's set up.
Okay. There's a question from Rory, kind of a comment from Rory, and then a question that he's been following the ACP for 10 years or more, have seen lots of technical presentations and research papers, PhD presentations, etc. Can I ask then, this is the question, has the ACP produced any accessible guidance documents, best practice guides, list of mitigation measures for farmers and landowners? And if so, where are these best practice guides available?
Yeah, so we have a dedicated website on the Teagasc website. So there's a page dedicated to publications, and they're there. And we have a KT specialist now. So Eddie Burgess does a lot of outreach work with groups. So I'm sure he can be contacted as well.
Okay.
For a follow-up.
Okay. All right, for Oggy then, from Coleman, were there any background nutrient tests done, for example, of non-water soluble locked up P levels?
Sorry, can you repeat again?
Were there any background tests done for non-soluble P levels locked up, I think?
Well, when we're talking about the P fractions in soil, and I'm just talking about soil because I'm a soil scientist, and then my colleagues would be more from the hydrology side and would be in the water quality. So not sure what is meant by non-soluble forms, because when we're talking about the forms directly available to plants, which would be the bioavailable phosphorus, that's only like less than 1% fraction of the total phosphorus, and the remaining phosphorus, more than 99%, is locked up in soil.
Okay. Okay, and another question then from Brendan. So he's just picking up on the fact that 20% of the soils have a index 4 P level, and that's above optimum, I guess, for agricultural growth. How can we address this, or maybe what steps are being taken through the Agricultural Catchments Programme to address this, to, I suppose, to reduce the level of soils at index 4 P?
Yeah. What we're focused on, and it has been like that since the first results came in the Catchments Programme in relation to phosphorus. And it has been mentioned already that we have an advisor working with all the farmers within the catchments areas. And what we could do, and what would probably be the best thing to do in terms to mitigate the phosphorus losses to water, is first to, when we're talking about the P index system, and so to avoid any source of P as a fertilizer on the areas which are P index 4. And if within our farm we have areas, as I shown in the last slide I had there, so there would be like either areas used for silage, or some areas like part of the milking platform with the lower P content. And especially when we're talking about organic manures, like there's a significant potential in organic manures, and we tend to forget that organic manures, whether that be slurry or some other organic fertilizer like poultry manure or pig slurry or anaerobic digesti, they contain a significant amount of phosphorus. And if we redirect that organic manure to the fields with the lower P index and avoid P application in the fields with the P index 4, that would be, that's in the context of our nutrient management plans, that would be something to mitigate P emissions. And then the other thing we could do is it's important to know, well, the most kind of risk in terms of phosphorus pollution comes with the so-called critical source areas. So if our fields with excessive P content are hydrologically connected to the nearby water bodies, these are the areas which present the biggest risk. And if we try to break the pathway of that water, like coming into the stream by implementing either, I don't know, some sort of buffer strips or riparian buffers or anything, or just to keep the soil under the permanent soil cover, like to have it under permanent grasslands, or to use catch crops in terms of tillage crops, that would, like every blade of grass actually slows down the movement of water and helps in a way to mitigate the P pollution.
Augie and Bridget, sorry Tom, were you going to come again? Or
no, I was just going to mention one comment in the questions just from Kieran, I think he's our colleague in Teagasc, and he just says that index 4 soils, it's going to be very difficult to reduce the percentage of index 4 soils where the slurry is being put out on the milking platform. So I was going to finish by saying that to deliver on what Augie has outlined as a practice to bring about change, the farmer has to make decisions, different decisions to the decisions that are currently being made, and that requires a change of practice. So that's it.
Tom, thank you very much for giving us a dig out of the questions. Bridget and Augie, thank you for taking the time out of a very busy week, and the best of luck with your open day today. I'd just like to conclude by reminding people, next week we have Dr. Sarah Kavanagh, and she's a project manager on an EIP project that's specifically looking at protecting farmland pollinators. I'd also like to thank our colleague Yvonne behind the scenes, who does all the technical work for us every Friday morning. And thank you all once again for joining us, and hope you can join us next week. So until next week, slán. Bye-bye.
All right, thank you.